So I'm Becky Sappington, moderator for session 3, which is on AI and imaging. We have a fantastic panel, and I'll introduce each of them when it's time for their talk, but we do want to provide the learning objective, so why we put this session together. So the goal is to evaluate imaging modalities for eye-brain connectivity in glaucoma, recognize opportunities to employ AI for preclinical outcome measures in glaucoma, in addition to what we often think about with AI and imaging. And then our final talk is going to speak to how you can collect data on the front end that is AI ready. So we all know there's a big emphasis on AI use in glaucoma and glaucoma funding, or all research funding. And so I think this is going to be a great session. So with no further ado, I'm going to turn it over to Kevin Chan from Stanford. There we go. Yes, he's going to give us an overview of imaging, OCT, and MRI.
Thank you, Becky. Testing. Yep, and thank you, organizers, for having me, and thank you, everyone, for coming over. So today we would like to discuss about one of the major clinical challenge, actually. And first of all, I have no disclosure. The clinical problem is that we have pretty good and effective IOP lowering measures, but IOP is actually the current only clinically modifiable risk factor for glaucoma interventions. Glaucoma is, for most of the case, irreversible, is a neurodegenerative disease of the visual system. In quite some cases, even after reducing eye pressure to a normal level, it may still continue to occur and progress. So we actually are very eager to identify, because of the irreversibility, some early markers for detecting glaucoma. And because IOP is currently the only methods for intervention clinically, can we also find other potentially important factors that may be druggable and more specific to slow down and decrease the severity of damage caused to the eye pressure, visual system beyond eye pressure. So in the next 10 minutes-ish, we would like to kind of navigate together around in vivo non-invasive imaging biomarkers for glaucoma, specifically for patients. We want to focus on the OCT for the eye imaging in patients, and also the eye is actually the main organ for sensing, receiving light signals, but it's actually the brain, almost 1/3 of the brain, that is interpreting and receiving these sensory signals to interpret and understand the world, the visual world. So we also want to know how the brain is actually working with the eye when we have glaucoma, and ultimately how the eye and brain are actually connected together, and are they interacting in addition to degeneration? Is there something else happening? So with that, to start with OCT, in the current background of the clinical standards, I think our opening talk from Dr. Yvonne Wu already mentioned that glaucoma typically involves the retinal ganglion cells. And fortunately, in OCT, we can actually evaluate different types and aspects of the retinal ganglion cell. For example, for the axons of the RGCs can be found and detected in the retinal nerve fiber layers around the optic nerve head towards the fovea. And there are also contrasts available below the RNFL. For example, there's the cell bodies of the RGCs called the ganglion cell layer. And then after the GCL, there's also dendrites of the retinal ganglion cell in the placiform layer. And OCT has this capability to help us understand the thickness upon proper segmentation, and of course good quality acquisition. So the question here is how early can we actually detect glaucomatous changes, or if there's a patient with these biomarkers, can we be able to probe into their changes early enough so that we can slow down disease progression in a safe fashion? So one of the current standard ways to evaluate glaucoma in the clinic is to use the OCT reports. For example, one of the most popular report is to examine the optic nerve head cup-to-disc ratio and also the neuroretinal rim. In addition, you can also evaluate the peripapillary RNFL using this report. You can see there's a overall thickness map of each eye. It's relatively difficult to just look into the map's thickness to evaluate if someone has potential glaucoma or progression. So this report actually offers a deviation map that is computed against the age-matched normal population, so that it may give you some hints if there is any characteristics patterns of glaucomatous changes in the patients. And you can also evaluate the circular measurements around the neuroretinal rim and also the RNFLs, as previously discussed in opening talk. In terms of how early we can detect it, so that you can actually use some available clinical visual field measures to compare against RNFL. For example, if you plot the visual field clinical measurements against RNFL, you may notice that there's not really just a linear trend, but instead you may notice here that RNFL may start to decrease in like a plateau fashion, a slope, without much change in the clinical visual field until reaching a tipping point. In this case, this is 76 micrometer. Then only after that point, then you will see concurrent RNFL and also visual field worsening. So this is one way to kind of suggest that maybe there are some potential rooms that we can detect changes in the retinal thicknesses before there's a clinically detectable visual field. And there are also some other follow-up studies suggesting that not only RNFL, if you look into the neuroretinal rim, there's also similar patterns, suggesting maybe you have some ways to probe and modulate these changes before we could detect further vision loss. So RNFL is one of the reports. There are also complementary reports, such as the macular ganglion cell in a placiform layer, we call it GCIPL, is occurring in quite some clinic and research institute already. Part of rationale is that you can, in addition to looking into the axons of the RGCs, you could also evaluate like the ganglion cell, soma overall thickness, and also the dendrites in the inner placiform layer. And so there are some suggestions that perhaps GCIPL could be earlier or more sensitive for detecting early disease, or maybe in RNFL, because there's a flow effect for RNFL in advanced glaucoma, it would be harder to detect advanced glaucoma progression. So GCIPL could also be an alternative for evaluating both early and late effects. To really quantify how they may be altered across these markers, you can actually plot, for example, RNFL and GCIPL, and also cup-to-disc ratio against the clinical field deviation. And you can compare how much of the percentage loss may be occurring before you can reach the tipping point. And in this case, using same cohort, you notice for RNFL, it will require around 14% loss from the healthy group before reaching tipping point. GCIPL in the macula is about 24%, and then for cup-to-disc about 99%. That said, it's not really direct reflectance of which one is occurring early. It's more like it's reflecting the current sensitivity of this instrumentation. And there are certainly some ways you can further improve, like the precision. Here in the GCIPL, we are talking about combined ganglion cell and inner placiform layer, partly because of the intrinsic limited contrast between these layers. So some of the current machines that are trying to tackle these methods include using, instead of the conventional near-infrared light, there's also visible light, OCT, using shorter wavelength for high resolution because of the short wavelength, and also these biological tissues can also have a better absorbance, so you have a better contrast. Especially here, you can differentiate between the GC and the IPL, and not just that, within the IPL, there's also possibility you can further look into the sublayers that may be sensitive to like the on and off of the RGCs. And for other methods that could further enhance the sensitivity for RNFL, Professor Chris Liang from Hong Kong also developed optical texture analysis of the RNFL using algorithms including both thickness and also reflectance and also some AI-powered analysis. So you can see that in the conventional RNFL and GCIPL, you don't really see much of the deviations, but using the rotor methods, you may notice there are some dark aqueous pathway that suggests being more sensitive to detect. The same is for the second subjects. Showing on the top, there seems to be some kind of obvious contrast, but in the deviation map in conventional report, we don't really see much. In terms of the optic nerve head, this is an AI session, so we were also interested in not just classifying, but potentially understanding using AI. Can we explain or interpret what may be the possible features in contributing to the classification between glaucoma and maybe healthy subjects? And this is an optic nerve head OCT, and you can actually use some kind of explainable, interpretable AI, like a classification map, to understand perhaps the optic nerve head near the cup to this, that this area seems to be contributing quite strongly to the glaucoma versus healthy contributions. And beyond that, you may also notice there's a cluster deep down. This area is actually close to the lamina cribosa. And if you use some other techniques like AOCT or enhanced depth imaging or swept source OCT, you may be also be able to look further into these beams and pores near the lamina cribosa. This is one of the major sites of injuries that people are really actively looking into how those beams and pores interact, if there's high pressure or if there's any other potentially non-IOP related factors contributing to these areas. So in terms of the brain imaging, we are very curious to know, since there's potentially early changes you can use in OCT for the eye, how about the brain? Are there also early changes? And one way is to use the function MRI by probing into oxygenation in the blood. And this blood oxygenation is also carrying oxy versus deoxyhemoglobin, and deoxyhemoglobin has a heme being exposed into environment. It will give you some negative distorted contrast. So that way you can give a visual stimulation to the top or to the lower field, and then you can actually evaluate the amount of the blood oxygenation related activity. Here you may notice that upon different types of visual field stimulation with increasing severity of glaucoma stages, there seems to be relatively weaker and weaker functional activity being attributed and recruited. This suggests that maybe a weaker brain functional response is happening with increasing severity. And with that said, is it also showing any potential tipping points or early changes before visual field? And we found that this is potentially the case too, using the same patients we just showed for the RNFL, GCIPL, and optic nerve head. There seems to be also some potentials you would be changing brain functional activity while the clinical visual field function remains relatively undetected or stable. So apparently there are quite some rooms you can really try to look for ways to intervene during these periods. And that said, you can argue, because this is a functional response towards the outcome or output. It could be contributed by the eye, it could be contributed by the brain locally. So what is actually happening within the brain in addition to the eye? And MRI is really looking multimodal, so you can evaluate, for example, using the water diffusion to probe the water microstructure environment. And here you may notice that in the warm color, this is an early glaucoma group versus healthy control groups, which suggests that not only in the optic tract, which is connected to the retinal ganglion cells, suggesting damage along the axons, there's also optic radiations which require transsynaptic transport. That is also structurally disconnected or disorganized, which may suggest there's also transneuronal or transsynaptic degeneration. Beyond that, there's also some areas called SLF, at the superior longitudinal fasciculus. This area is actually playing a lot of roles in the multi-sensory integrations. So we also observe some sort of structural disorganization, and that may potentially attribute to some kind of a multi-sensory integration. We're speculating if this is related to, or if you can explain, for example, if glaucoma patients may have high risk of falls. Maybe it's not just an eye input deficits. Maybe there's really something in terms of visual motor coordinations, and this is ongoing work. So now we have eye imaging, brain imaging. So how does, yeah, okay, so no, that will wrap up, no worries.
I don't know how many slides you have left, but.
Yep, it would be quick. Thank you. Yep. So this is near the end. We want to actually answer some questions that was hypothesized like 10 years ago, suggesting that when you have actually the visual field from each eye, very often statistically, the left and the right visual fields, they may be not identical. They may be actually complementary. For example, in one subject here, the visual field in one eye is deficit in the lower visual inferior regions versus the superior regions. And this seems to be beyond random in their analysis, and they suggest that there's a potential brain control of the neurodegeneration, and that may also explain why one of the questions in the audience suggesting why the patients are hard to sense that they have glaucoma because they're using both eyes to look together. And they're starting to have some scratches about what may be causing this, or at least associated with that. For example, we can use those biomarkers from the optical radiations and microstructural environments. We do a partial correlation with 62 subjects, and it turns out that in the left eye, the intra-axonal space water environment and diffusion tend to be more associated with the upper visual field. And then right eye is more in the opposite field. Extra-axonal space is more related to the myelination, inflammation. One eye seems to be more in the central vision, the other is more in the periphery. Beyond that, you can also look further into like spectroscopy of the metabolism, and it's also very curious to see that we can examine the visual cortex GABA, which kind of associate with the upper visual field, whereas the glutamate and choline is more associated towards the lower visual field. Last but not least, many of us are looking into the image formation to see better with the environment, but there's actually the non-image forming visual pathway as well, which is kind of driven by the intrinsically photosensitive retinal ganglion cell. And we have tried to look into the connections called the retinophthalmic tract in our patients. This tract actually plays a lot of roles, like in autonomic nervous system, eye pressure, blood pressure, circadian rhythm, energy balance, and metabolism. And there are some nuclei that are known to be connected to these hypothalamic regions. Looking into our patients, when we segment them into 5 major areas, we noticed that there is actually towards the lower part of the hypothalamus that will show more decrease in the volumes. And all these decrease in volume towards disease stage is also linked to like blood pressure, linked to retinal thickness. And there's one region that's no volume change, but it's related to eye pressure too. So are these in the future potentially relate to a role of something more than vision? For example, these hypothalamus is playing some role in sleep, and there may be also linkage to anxiety and also stress. Can we be able to probe into these potentially modifiable factors beyond eye pressure? And other questions include can we also look into like normal tension glaucoma mechanisms by probing into the CSF, cerebrospinal fluid dynamics, and also the vasculature, the OCT that Dr. Fortune already touched upon. All these could be potentially looked into as an integrated process. And finally, trying to link to the previous talks on the modeling, using AI, there's a lot of AI modeling on humans. What if we also try to leverage the AI from the human side towards the preclinical end? Can we, for example, leverage them for rodent OCT using transfer learning? We have some collaboration with our moderator on that. So this is our take-home message. We believe that these in vivo live imaging markers can offer opportunities for early and detection and widespread changes in the visual system. Hopefully we can help improve the detection and monitoring of disease processes. Maybe we can translate between preclinical and clinical research, and also facilitate ways to identify and test strategies beyond IOP measurements. Thank you very much.
Okay, next we have Luca Della Santina, who's going to talk to us about AI approaches to identifying synapses, and he's currently at the University of Houston.
Hello, everyone. So the task that I have today is giving you a good idea of, on a single cellular level, how we can, or what's the role of AI in helping us identify synapses and potential changes that happens during glaucoma. The only disclosure that I have to make is that I am the inventor of one of the programs that I'm going to talk on today, but I don't have any associated financial benefit from it because I made it open source for everyone, so everyone is encouraged to use it for free and modify it and contribute to the field. So many of the pathological problems associated with neurodegenerative diseases like glaucoma are due to the fact that we either lose neurons, or maybe we don't lose them, but we lose connectivity between neurons. And every time that you have a synaptic dense neuropil like the retina, you have possibility of loss, or some of you might be interested in recreating additional neuron. Well, these neurons had to integrate with an existing network of synapses. So the role of AI here is helping some of the fundamental tasks that we have during image analysis of synapses. So we're going to look at synaptic labeling and imaging, and we're going to look at how we can segment these images and validate the presence or not of synapses. Two main advantages can come from integrating AI in this workflow. One is to expand our area of investigation. So normally we used to look at very small portion of the retina or the brain. With AI, we can potentially multiplex this to the entire tissue. And second potential advantage is generating consistency in the way we analyze the synaptic data, either between different labs or across time points in a longitudinal data set. Fast forward to the 2010, 2020s, we have two good ways to image and label synapses. One is using electron microscopy, in particular either serial section or serial block phase EM. Back in the days, a lot of time was spent aligning EM images. Now this is solved by the engineering part. So we can generate very good quality and reliably aligned images of retina stack in this case. And you can see here that the next task is really identifying in these images where the synapses are, how many they are, what's their shape. And this exposed a lot of diversity even within the retinal synapses. You see here the all possible synaptic motif of just bipolar cell axon terminals. So we, in the retina, differently from most of the rest of the brain, we have the distinction between either synapses that have a ribbon or synapses that are ribbonless. And within ribbon synapses, we have synapses that are with multiple partner, with one partner, and even more, we have same synapses that could allocate more than one ribbon to the same synapse. So there is a lot of potential for the AI to help with the classification of different types of synapses. The other way that you have available to label synapses is using fluorescent markers. You can do this leveraging the different specialized proteins that are on the two sides of a synapse. We can separate different types of synapses depending on their structural anchor protein or proteins that load the vesicle with the neurotransmitter. And we can do this by doing immunolabeling with the whole tissue. That makes it easy for tissues that otherwise are hard to transfect, for example. Or we can transfect the protein of interest fused with a fluorescent protein and then look it up afterwards. And This approach leverages on fluorescence microscopy, and in the latest years, we had massive expansion of the ability to generate large volume data set that contain synaptic information. Once you have the images, and that is many times a very time-limiting factor, then you have to make sense of these images. And this is when AI becomes more and more useful. If you work with electron microscopy, this is a typical workflow that involves AI. One of the first segmentations is to segment the membranes of the neurons that are connected with each other. This can be helped by AI by using pixel classifier or semantic segmentation networks. And once you have this initial reconstruction of the neurons, then you can also identify the point of contacts between the neuron. And if they have the hallmarks of a synapse, like a ribbon or synaptic vesicles or a postsynaptic density, then you get additional clue that that's actual synapse to look in your tissue. If you look in a fluorescence microscopy, this is an example of a transfected retinal ganglion cell that is also transfected with a second plasmid that express PSD95, so a postsynaptic protein for excitatory synapses labeled with fluorescent protein. And this allows you to have individual highlights of where the postsynaptic sites of a given excitatory synapses is, and we can use image segmentation algorithms to segment the shape of the neuron. This really doesn't need AI because we have pretty decent graph methods and iterative thresholding algorithms to do this. But then you get to the point that you have to recognize each individual synaptic puncta, and this is where AI can actually help with the decision process of what's the boundary of an actual synapse and where one synapse is separated from the next one. Once you have segmented your volume of interest, then the next step is validating these potential synapses. And at the end of the day, most of the image analysis that involves synaptic quantification involves one of these three cases. One is looking at synapses that are within a cell of interest. Think yourself about looking at the axons of amacrine or bipolar cells in the inner retina. You could be taking an approach that is more agnostic of the cell type and looking at every possible synapse and how it's aligned with pre- and postsynaptic sites. Or you could just look at the dendrites, for example, of a ganglion cell that is in glaucoma and look exactly where the input synapses are. So this is, I'm trying to exemplify for the retina, but this really applies even if you're interested to study the recipient part of the retina. So the axons of the ganglion cell and the LGN, for example, have a similar problem to solve. And there are many ways that you could validate your synaptic data set. Some of the most useful one is using multiple imaging modality to make sure that what you're actually considering a synapse is a synapse. For example, you could do fluorescence imaging, then retrieve the sample and do serial EM and double check that really where I see a fluorescent puncta, there is a PSD and a synaptic cleft. The other possibility is looking at a position between pre- and postsynaptic markers. So if we know that this is an excitatory synapse, we can label for specific pre- and postsynaptic proteins that we know they are at this synapse, and we can look at a position of these two proteins. And the third way that is a good way to validate the synaptic data set is looking at two proteins that are supposed to label the same side of the synapse. So that will also validate your data set. So for example, this is the workflow for using AI doing EM imaging. This is done with Synaptic Connectivity 2 program. You see here you have an initial neuronal segmentation of the EM data set, and after you have that, you have identification of potential synaptic contact points and final segmentation of the whole image. This is a graph that shows the time cost of doing all this computation given the volume that you're trying to analyze. And you can appreciate there is different tasks, but each of these has either a linear or a sublinear relationship with the size of the volume that you want to look. So this is basically the time you can expect to analyze this data set. If we do the same task with fluorescence, the advantage of this approach is that normally you can generate the images much faster, but you also have much more data to analyze. This is how we have Object Finder doing the job of validating synapses, so we can extract each individual synaptic candidate, generate projections in the three fundamental planes, and then use a deep learning binary classifier to detect whether this is likely to be a synapse versus a noise or a sediment in the tissue, for example. The main advantage of doing this is that in terms of cost to process the tissue, the time that we need to do this classification is actually proportional to the logarithm of the amount of synapses you have to find. So it's way faster than we would do with the EM. So if you want to look at a very large data set, this is still probably your best shot to do it in a reasonable time. In summary, the most use for the AI is either in the segmentation step, and this is extremely true for EM, or in the classification step. So you can classify different types of synapses from each other using AI, and this is true for both the EM and fluorescence images. We have, at this point, multiple open source tools that we can use for this. These are only some that are the most updated. So Object Finder, Synaptic Quantist, and Bot for fluorescence imaging. And then most of the retinal connectomic people use Knossos or VAST for EM segmentation. A couple of pitfalls to avoid. So first important pitfall to avoid is don't just use whatever model is published. Just always make sure that it's fine-tuned with your own lab data, or at least you know what's the performance of that model on your own data. Second important pitfall is avoid training this model on an extremely uniform data set. Variety in the data set allows the network to generalize. So if you don't do this in the training step, you lose that property. And third thing is make sure that what you use to validate your model is actually representative of the data that you will use the model for. Okay. A few rationale on how to use a good model for synaptic analysis. The first one is if you can, you should probably use an open source model so that you know exactly what's the rationale behind the algorithm that you're using, as opposed to use closed source proprietary software in which we don't know actually what the algorithm does on the back of the house. Second thing is favor models that come with the training data and the parameter that is being used to train the model. This allows reproducibility, at least, of the data that it comes with. And third important thing, favor models that are trained on data that is similar to the one that you want to use. So the amount of retraining for the model is minimal. A few unanswered questions that eventually our field, like the rest of the connectomic field, has to figure out. One is, can we reuse some of the models that have been developed in the brain for identifying synapses in the retina? For example, a lot of excitatory neurons in the brain are spiny neurons, but we don't have that in the retina, so at least that involves major retraining. Second important thing is how robust are these models that we may train with control data to actually identify data that comes from diseased tissue, like glaucoma tissue. So I'm going to give you a few more information in the next talk of Friday about that. And then third thing is, can we use the approaches like Kevin showed of explainable AI to actually dig into the rationale that the model itself does in the decision process to say, okay, this is a excitatory versus inhibitory synapse. These are some references. I divided them between fluorescence and electron microscopy. And also, these are some of the open source software that you guys can download and use right now. Thank you.
Thank you. Next up, we have Mike Anderson from the University of Iowa, and he's going to talk to us about use of AI in counting axons.
Hi, it's a real pleasure to get to talk today. I'm very thankful for the opportunity. This has become one of my favorite meetings, and it's a real pleasure to see so many of my favorite colleagues here, and also all the new faces of people who I hope to get to know in days and years to come. So I'm going to start my talk today, actually, by saying I don't have any disclosures, but then with an analogy. With a room full of curious and confident scientists, I know that this will be one that resonates. Have you ever had something in your house that stopped working and you look at it and you think it can't be that complex, I'm going to go fix that. So off you go to go get the fancy little screwdriver and you take the 4 little screws on the outside off, and 3 minutes later the screws are all lost, your kitchen table is full of gears, circuit boards, and who knows what's that are never going to go back to the thing again. Well, that's exactly what axon counting is. It looks pretty simple at the surface, and once you get into it you see that there's a lot more to it. And what I hope to do today is to give you a few tips, things that'll get you started going in the right direction and make your life a little bit easier, and also make you alert to some of the things that you can expect once you take the cover off of it and get into it. So switching from my microwave to axons in the optic nerve, if you're going to count, at the end of the day, ganglion cells are what die in glaucoma, and so you need to either count their somas by doing flat mounts in immunochemistry typically, or by counting axons in the optic nerve. Typically, those have a one-to-one relationship. There can be exceptions. For instance, with BAX or BCL, you can have a soma without an axon. With wall iron degeneration slow mutants, you can have an axon briefly without a soma. But normally, these things are one-to-one. In glaucoma, it's been found repeatedly that the axonopathy tends to precede the soma cell loss. So if you want a more sensitive assay for early stages of disease, many of us end up looking at axons. If you're going to do that, luckily, you can lean on a couple of techniques. Axons are small and they're soft, so you have to do plastic histology. But there's been techniques worked out to label the axons to make them visible. One of them is by using this paraphenyl PPD. In this technique, you have to use osmium-fixed tissue. So osmium binds to the lipids that are in the tissue. PPD chelates the osmium and thus makes it dark. So it was developed decades ago for people doing TEM who wanted to be able to do light microscopy on their TEM samples at the same time. But the PPD ends up staining the myelin, which is a lipid. And so if you're going to do that on an optic nerve, you typically start with the whole cross-section, take a sampled area, and when you start to look at the axons, healthy axons show up as donuts, basically. So the PPD just stains the outside of the axon. Damaged axons show up as solid. So the axoplasm stains the PPD when they become damaged. And then the empty spaces are basically the glial nuclei and processes. And because the glia are kind of like squid, so they've got lots of appendages that go all over the place, in a cross-section, these are only micron-thick plastic sections. In a cross-section, you catch the nuclei once in a while, and the processes are basically just the tracks where you don't see any staining. But the glia do take up 20 or 30% of the total cross-sectional area, so there's quite a few of them. So then looking across the disease state, what you find in those 3 things, in healthy optic nerves, you find the healthy appearing axons, just a lot of donuts and glia. As the disease progresses, you have fewer healthy axons, more sick axons, and the glial processes tend to take up more space. You also come up with some amorphic material that's we've referred to variously as things like gliotic scars. It's who knows what exactly. None of the above, as you see in that image of the severe disease. You also have the option of using toluidine blue, which lots of papers will do. It's a very similar thing. Toluidine blue binds to acidic components in cells, and the oligodendrocyte processes that enwrap the axons, and actually it's their plasma membrane that is the myelin sheath, they bind toluidine blue. So toluidine blue in effect does the same thing as PPD. It shows you where the myelin is. And the healthy axons, again, you just see the myelin. And The ones that have a thicker thicker myelin sheath around them are ascribed to being the damaged axons. So you'd think it'd be really simple. We all know how to count. You should be able to just sit down and count the circles, and that would be the end of it. This is where sort of the cover comes off, and you're like, oh, geez, there's a lot more to this. So some of the anatomical challenges to counting axons are shown here. First, axons, they're small, so they go from about 0.3 microns to 20 times that, 6 microns in diameter. There's a huge number of them. So in human, it's around 1.1 million. But that number actually varies person to person. It can be as low as 700,000, as much as 1.5 million, thereabouts. In mouse, it's about 45,000. But again, depending on what kind of strain you're working with, it can go from 30,000 to 90,000. And there's about a 4% variability mouse to mouse within an inbred strain. So there's some environmental noise, some flux in this machine, that there's a lot of variability. So you're counting a lot of very small things. And then as you saw, the disease heterogeneity in both instances, the optic nerve appears different, and it's harder and harder to find what's exactly an axon. There's also workflow challenges. Raise your hand if you've counted axons before. Yeah, that's what I thought, almost everyone. You preaching to the choir. And this is hard, way harder than it looks, than you would think. And one of the challenges is, so in my lab, we try to count about 10% of the optic nerve cross-sectional area, and one person can do one nerve in about 1 day. So if you've got a data set of 20 controls and 20 experimentals, you're looking at a long project to get all those things quantified. If you're like me, you're probably looking at that image and saying, why did he count that an axon and not that? There's person-to-person variability in this. So despite how well you train people, you end up with a little bit of noise in how you end up counting them. But right now, what you would do is you would take that time and just do your best, and you'd take the density times the area and call that the number, and that's what we tend to go with. So some of the needs that we need to get to in the field are to do whole nerve sampling. We need better workflow that's faster. The two biggest things are probably we need more reproducibility. The two labs should be able to do the same experiment, get the same result, and then rigor, just by having more quantitative data. But also with AI, we'd have the opportunity to do things that today we can't do at all. So instead of counting one section per optic nerve, why don't we count 3 or 5? All the cost is in getting the slide made. Once you have it, if it's easy to quantify, we could do 3 and average that to come up with a really accurate number per mouse of what the exact number of axons are. So I'm going to walk through 3 of the different approaches that exist right now for how to use AI. In all of these approaches, the fundamental thing is that you have a human segment where axons are and some subset of images. You call that the ground truth, and then you use encoders that go through and sort of look for different features to make a feature map. So things like, where's there a horizontal line, where there's a vertical line, et cetera, et cetera, et cetera. And then the decoder part of the AI is what calls, this is the edge of the axon, or this is the myelin sheath. AxoNet was developed largely by Ross Ethier. A few of the unique features of this were that it used, there were 14 people that helped segment the training images, and they took the average of all of those to make sort of a probability map of where the axons were. They used 1,400 sub-images that were relatively small, so they used a lot of small images. And these were done, it was trained to recognize healthy-looking axons from rats that had a variety of damage levels. A really important part to all of these models that I'm going to talk about today is that they were developed using succinct subsets for training, validation, and then testing, because it's really important early on to make sure that you don't overtrain your model to just recognize what you're training it on. So at the start of the experiment, you have to put aside a certain fraction of your data and say, we're not going to touch that until it's time to test it. And that's one of the stringent things that the better models will all have. So in this instance, what they came up with then is shown here. They end up with an AI-based approach that had a performance similar to human. Highlighted in red there, it's showing DICE scores. DICE is a number that's meant to show the similarity between 2 images. And so the higher the number, the better it is. What you can see is that the performance between 2 human annotators, the DICE score is 0.87, and the axonet versus human was 0.81. So that was a really good performance. One of the things to tell you is that this is available on GitHub, and it has a guide with test images, so it's relatively easy to use. I would caution you to use Anaconda, so you probably need some IT to help get that installed on your computer, and you might need some help getting that to run. But the guide walks you through it fairly straightforward and gives you annotations that work. Second model that I'll tell you about is called Axon Deep. This is one that I worked on with Mona Garvin at the University of Iowa. It's similar in that we had graders, and the first thing we did was we graded the nerves, 1, 2, 3, mild to severe, then split them up into our training sets so that our training sets and our validation set and our test set had similar representations of damage levels between them. We only had 2 graders, and we used fewer images, but they were very large images. The thing that was a little bit different about ours was in the AI part of it, we used an adversarial network. So this is an AI kind of one of the many flavors of AI that you can do. There were 2 networks. One's a generator and one's a discriminator, and you try to pair them in a game against each other. The discriminator is making up fake images, and the discriminator is trying to tell what's fake and what's not fake. And you keep doing that reiteratively until you end up with a situation where it can't tell. The one that's making up the fake images can do it better than the discriminator can tell. That's basically gives you your answer. This is how the AI can figure out what an axon is. The other thing that was unique about using that approach is we're trying to utilize unlabeled data. So in that approach, you don't necessarily have to have one person trace a gazillion axons. We only used a fraction of the data that was actually manually annotated. And by this adversarial network approach, we were able to make use of the unlabeled data to help learn. And what we found, shown here, we compared it to a full convolutional network, traditional, you circle every single one that you want to count. And we compared it to this semi-supervised approach with adversarial networks. And what you can see circled in red is by using 50% or 25% of the data, we were able to get almost equal performance. So this is a way that you could train a network, for instance, to not just recognize healthy axons, but damaged axons, or anything else that you wanted to try to teach it to learn, like glial processes. And I would warn you, this method is not consumer packaged. So this is meant as a methodology. It would take an electrical engineer to implement it, or calling Mona or I to actually run it right now. We're working on trying to get it packaged. Third, I'll tell you about fine-tuning foundational models. So with AI, you can actually sort of borrow the encoder from other approaches. So these are 2 things that were made. RET found was made to segment fundus images, which we can actually take the encoder from it, instead tune it to recognize axons, and get relatively high performance levels. One of the summaries is that these models are not yet perfect. Where they do well is on nerves with mild to moderate levels of damage. Where they do poor is on moderate to severe levels of damage. So this is just one example from our lab. Where on the top you have mild, and then moderate, and then severe on the bottom. We're comparing our manual counts to these applications of these 2 approaches. And you see as the damage level gets higher, the performance goes down. One of the pitfalls is that some old challenges still remain. One is, where do you sample from? If you have to do sampling instead of whole nerve, if you send an undergraduate who's not been trained to do counting and take images for you, they'll come back with images of the optic nerve that have nothing but axons and are avoiding all the glia. Same thing, you still need to deal with that. It's also possible to have to have regional damage in an optic nerve. This is a nerve from traumatic brain injury, but the same thing could happen in glaucoma, where you could have only a fraction of the nerve that was damaged and a fraction that was not. And imaging a whole nerve continues to be sort of a challenge, how you get a good full image. So these tools are useful but not perfect. It can provide quantitative outputs and improve our phenotyping. The future directions where I think the field will be going will be improved performance in states of damage, simultaneous segmentation of multiple features like healthy, damaged, and glial all at the same time, and convenient sharing of the resource. Thanks.
Thank you very much, Mike. There we go.
And our final speaker in the session is Cecilia Lee, who's now at the Washington University in St. Louis, but previously was at the University of Washington, so you may know her work from there. And she's going to talk to us about AI-powered insights from clinical data and how that can advance glaucoma research.
Good afternoon, and thank you so much for this real privilege to be here. And then I was really excited to meet a lot of people, and I hope I get to meet a lot more. Okay, so I have some grant support, but none of them directly related to this. I'm going to just share 2 examples of clinical research, and I'll mention AI Ready study and then end the talk with future directions. So I think Dr. Yvonne really said it beautifully in terms of, she spent an hour talking about what is glaucoma, right? And then, the clinical standard or current standard of, at least clinically in our clinic, we are trying, really hard to really define and then kind of categorize or, grade the severity of cup to disc ratio, right? And then so we're looking really carefully with 90 diopter lens or, color fundus photo, and then, or this photo. Or, we are looking at the side-by-side, looking at HVF, Humphrey visual field, and then, I'm kind of like scratching my head, what did the progression happen? Is that like, artifact? Was the patient kind of sleeping and, dozing off? And, so you're trying to kind of really diagnose that, and then because you have to make a management decision, right? And we wanted to try a model where we could just give one Humphrey visual field, a baseline Humphrey visual field, and then say, what do you think that Humphrey visual field will show me in 5 years? And then that was kind of our goal. And instead of us try to really say, oh, Dr. Lee, can you say, did the progression happen or not, and then try to argue the definition, we decided that actually the aerial time is actually an undeniable ground truth. So you can't say, you can't compare 2 visual fields that are 2 years apart and say, well, which one is actually, the earlier one? Obviously, the later one is a progressed one, right? So then we trained our model, such as, hey, this is our input, and then 2.5 years later, this is our output. So then essentially, we are doing input, and then output, and deep learning, and then that was our basic model. So what do we do? So this was in collaboration with Joanne Wen, who's at Duke, is a glaucoma specialist at Duke, and Aaron Lee, who's at WashU right now. And then these are some of the examples of the extraction that we did from clinical Humphrey visual field at University of Washington. So this was the 24-2 Humphrey visual field that were obtained over 20 years of clinical practice at University of Washington from about 5,000 people. And then we obtained about 32,000 visual field, and then each visual field, we extracted all the perimetry points to the 100 decibels. So not just those numbers in the left side, but then kind of like those numeric values that you can get, the raw data on the right side. So we extracted about 1.7 million perimetry points. And again, the goal was to take a single Humphrey visual field and predict a future Humphrey visual field. And then all the input that they had was demographic variables and a Humphrey visual field. How do we do? So we actually trained a lot of models. I think we trained 8 different models. So fully convolutional neural net, and then also other deep learning models. And what I'm trying to show is that actually out of all the models, and then after 10-fold cross-validation, the model that behaved the best or performed the best was Cascade 5. But what I'm trying to show is that, the mouse, okay. And then so this is kind of our point of evaluation was looking at PMAE or pairwise mean absolute error. And then, obviously, they did pretty well in terms of the error rate. And then so this is like the Blunt-Altman plot in terms of where the errors in the actual Humphrey visual field end up. And then this is the error range as we go from early on to later on prediction. So clearly, yes, models having harder time in predicting what happens much later, right? Other than just a year or 2 years from now. That makes sense. And then what was really interesting was kind of the results. I was really amazed. One model was not tricked by saying, oh, this is not glaucoma, this is homonymous hemianopia, probably had a central CNS defect, right? Or had a stroke or whatnot. And then the other ones pretty good. I mean, 3.5 years, 2.3 years. So the model kind of recognizes already a baseline. This person is high risk based on the features of the Humphrey visual field at baseline, right? So what this tells us is that there are a lot of latent information that can help you to predict the future risk of progression just from the visual field, which may be not easily seen to us as clinicians at naked eyes. I mean, I know I can't do that. I'm not a glaucoma specialist, but I know I can't do that. So that was our first study in the glaucoma field. And the next study that I wanted to mention was that, again, Yvonne was mentioning about structure function. I think we really did hear a lot of structure function concept in glaucoma, and then that's what we do, right? We are trying to really evaluate the structure of the optic disc or, neurofiber layer on the disc evaluation, and then you're trying to kind of predict what is the functional loss or, risk of losing visual function in the future. And then we often do this. We are looking at the disc and then also OCT. A lot of talk was done on RNFL OCT data today. And really what we do is that, we again look at, there's an inferior thinning. And then so I'm predicting the superior visual field loss. And then also I'm looking at the, inferior thinning on the OCT. One of the things that we do, and then so this lag between the clinical practice and the model development, what we do in the data science lab, is that there's a little bit of difference, right? As a doctor, as a clinician, you'll never just kind of see visual field and then hide your, and then like not look at your patient, right? You already immediately know, this is a woman, this is elderly female, right? And then I already know that, and then I have multiple modalities at my hand, such as physical exam on slit lamp. I have OCT, visual field. So I'm already doing this multimodal evaluation to deliver the best care to my patient. So that's kind of what we wanted to do. We wanted to kind of understand better what is this structural function relationship, and we can understand it better through the deep learning models. Garway heat map is a method where you can kind of translate a prediction model from structure to visual field. So you can use that from optic disc appearance to predicting visual field, or you can use that with OCT, the peripapillary OCT to visual field. So that's kind of what we did. We borrowed this idea. So this is the work of Yuka Kihara from our lab at WashU. And we said, you know what, we are going to train two multimodals. One, really using disc appearance from IR photos of the OCT, and then predict the visual field. And then another model is using OCT structural information and then predicting Humphrey visual field. And then why don't we fuse that and then use the best behaving model, and then let's see what helps us understand why that's behaving that way. So we developed two model and then fuse it later. And then I'm going to kind of show that the model is supposed to show us which input are you more confident, or are you really relying on to predict the output that I want? So for example, this little guy, I am using that visual field prediction based on the disc image. And then this guy, I'm using OCT data to predict the best image. And you can kind of see that if you are relying just on the OCT model or disc model, they don't do as well. This is the final visual field, the actual visual field. But then the final model that is combining both multimodal data, they do much better in predicting the visual field. And this was kind of the coolest part in terms of we were able to regenerate Garway heat map using OCT data alone, and then also using the disc photos alone. So I think this is kind of like an example of us understanding the mechanism, or the really reflecting what we do clinically, and then try to assess what that means throughout the objective model. I want to change gear. I know I don't have much time, but one of the big initiatives that we have right now in our lab is AI Ready. It's called AI Ready, an Exploratory Atlas for Diabetes Insight. So this is one of the major initiatives from NIH Bridge to AI program, where we have recognized the value or the potential of using AI or machine learning in healthcare research, but we lack a critical kind of ideal data set to study a lot, or to train a lot of downstream models. So we were one of the 4 funded studies project in the country, and our study was really to study salutogenesis or pathogenesis of type 2 diabetes. Why do I mention this type 2 diabetes one? Because this is a big data set that you as a glaucoma researcher can definitely leverage on. So what we try to do is really to balance the data set in the disease spectrum. So this is type 2 diabetes. So half of the people don't have diabetes. 1/4 of people are totally normal. 1/4 of them have pre-diabetic, and then 1/4 of them are medication controlled, and then 1/4 of them are insulin controlled. And we are equally dividing into Asian, Black, White, and Hispanic, and then men and women. And we go from toe to bottom. And this is a study that requires pre-exam survey, and then exam that lasts 3 to 5 hours, and then again 10-day course of environmental sensor, fitness tracking, and continuous glucose monitoring data set collection. So it's quite a bit. And maybe this will frighten you or this will excite you, but there are 44 different protocols that they have to go through from imaging. And this is an example of the data that we have already released. In year 2, last year, we released our data from 1,000 people. Our main goal is 4,000. And this data set alone, which is 1/4 of our anticipated total data set, it already has 43,000 macular and optic disc imaging, and then 6 million optical coherence tomography B-scan images, near 4 million OCTA flow B-scan images, and then, so you can take advantage of all these. And then if you are interested in kind of, what are the systemic risk factors of glaucoma prediction, then you have it. You have continuous glucose monitoring, environmental sensor, pollution data, fitness tracking, a lot of heart data. So this is all yours. You can actually download the data today. You don't need DUA. So I'm going to invite you to really look into this. Already 667 organizations worldwide have downloaded this data set as of September. And some of the images, we go from really cheap machine to really expensive machine, OCT, OCTA, and then we even have FLIO that are very research instrument. So this is yours, and year 3, containing data of about 2,200 people are expected to be released in mid-November. Take-home message. Longitudinal data enable robust AI models for glaucoma progression. And then we can really take advantage of all passively collected clinical data, as you have seen. Multimodal integration, such as visual field OCT or disc images, enhance structure-function mapping and predictive accuracy. And AI-powered biomarkers could transform patient care and our clinical trial designs in glaucoma in future. Some of the unanswered questions is, how do we validate these models across all the diverse population and health systems? And then also, you're going to say, oh, those models are great, but how do we actually integrate, and what are the challenges of integrating them in clinical practice? So I'm going to leave it here, and thank you so much. Thank you.
Okay, questions. Thank you all. Fantastic. Where's our mic runners? Okay, there you go. The mics are there on the corners. We have one in the center, one on the outside. You can go because you got your mic first.
I got it. So Mike, suppose you had a collaborator that wanted to work with you to count axons. Just suppose. What is the best form of data that you would want from that collaborator? Do you want the glass slides? Do you want like 100x images, oil? What do you want?
Well, first, you should really try AxoNet a little. It's not so bad. Even I could get it to download and run images. Honestly, it's one of the challenges in the field. Mona is scared to death that there'll be 1,000 people asking that question. And then my collaborator, Mona, who actually wrote all the stuff I spoke about, minus what Ross at the Air wrote about, how do you, short of making it a full-time job, how do you keep from having to answer that question 1,000 times, or having a place where you're storing these enormous images for X-known investigators? How do you deal with that image stuff? For us right now, for me, I could talk to you. We have space at the UI. We can store large images. 100x images are what we would want. And that's what Ross uses as well. But it is easier for the field if either our tools get mounted on the internet and you use them remotely, then you don't have to worry about updating your tool if Python gets updated or something, when Python gets updated. Or you download the tool and use it there, but then you have to incur more risk in informatics on your side. It's a real challenge, actually. Yeah.
I want to add a couple things. This is a problem of most of us. So this is a point in which foundations like BrightFocus or governmental funding agency can help with shared repositories that we can actually pull the data in, like we're doing for the deep learning that Cecilia explained, and we can actually all benefit from the same pool of data, basically.
A converse to that, Rob, is that we should really be making our segmented image sets publicly available better than we are right now. So we should all be posting those so that new folks out there working on AI and other fields might be able to easily take already segmented images that have been manually annotated and reuse them. Or yes, so that there's more marked data available for other people to use in evolution of the tools.
Brad.
Hi, Mike. I wanted to sort of underscore a really great point that you made about counting the whole cross-section of the nerve, because subsampling is potentially fraught when the disease is inhomogeneous. It's a basic Gaussian sampling problem. We have a paper a little over 10 years ago, TVST, where because we count 100% of the cross-section, we could do a bunch of simulations on subsampling. And it's really linear. The error goes up linearly with the less of the nerve that you sample, but more importantly, it gets further and further systematically off in the undercounting direction. This happens to be about age, but we know that age is not homogeneous. It tends to hit midget ganglion cells and papillomacular bundle more than the rest, et cetera, et cetera. Anyway, it's a really important point to build these kind of tools so that we can all count the whole nerve.
Yeah. Thanks for sharing that.
Applied any of these tools to retina flat mounts for RGC somas?
No, but use RGC code. There are other tools that count flat mounts pretty well. RGC code is the one to look for.
Coming back to that question of RGC flat mounts, I think it would be really nice to be correlative with RGC flat mounts and the optic nerve damage.
Oh, yeah.
You know.
Yeah, that would be the dream.
Yes.
Both eyes, both
Right.
tissues, yeah. Or one thing I didn't have time to say is, another thing about counting the optic nerve, though, is it saves the retina. So you could disease stage by using the optic nerve and then do your RNA-seq or your proteomics or metabolomics, or whatever it is, with the retina. So you don't have to use the tissue to stage it.
Actually ask a question, Cecilia. That was a great talk. I'm wondering, a lot of times in clinic when we see these visual fields on OCTs, they're not reliable or they're poor quality scans. How do these models take that into account? Are they able to somehow correct for that, or is that something that you guys are looking into?
So to be honest, a lot of models, when we train, we do get rid of poor quality data, right? And then And in theory, if you have big enough data with a various range of quality, then the model should be able to understand what the poor images are like, and then able to kind of do that. In reality, we don't really have a data set that's that big, and then also, we are trying to get, train the model with the best quality data. But I think using more instead of like RCT data, if you are really training using clinical health record data, the routine clinical data, then you are inherently using some of the quality variation within your data set. And then, you do a lot of kind of explanation, or, you do heat map, or you do, segmentation maps, and try to see where the data or where the signal is coming from. But it is a challenge in terms of understand or making a model that can take in any sort of input that has challenging qualities.
All right, thank you. I'd like to thank our speakers. I think, yes. Okay. I thought you were about to stop me for a second. Okay. Thank you so much. They'll be available for y'all to ask any additional questions if there are any out there. Thank you.